My name is Kemper, and my project is human immune cell responses to bacterial pathogens. So our immune system is basically there to fight bacteria and anything bad, and bacterial pathogens are like the bad bacteria. And the immune system has something called the macrophages, and they're basically there to engulf the bad bacteria, and it creates like, the macrophages are here, and then this infection or this bacteria comes in and they fight, and it creates this huge battle. So what Nedelec looked at was when this fight happens, this huge battle, what genes are being expressed, what is happening in this macrophage to fight back this bad bacteria. And so what they used was this RNA sequencing, which you can see right here. And the RNA sequencing basically has this RNA sample, and then uses the cDNA library and creates these reads, which are these long DNA fragments. And then we align it to a reference genome. Right here, you can see that there are a bunch of like reads, and then we have these genes, and that's how we get like the number of genes and how many times they're expressed based off these long DNA fragments. So what my project is, we used RNA sequencing to see if when there's two battles. There is when a macrophage and listeria come together, what immune responses we get, and then the other battle is when a macrophage and a salmonella come together, and then we compare them and see if there's similarities or differences. So what we did is we grabbed a bunch of data from the sequence reference archive, and then we did RNA sequencing it by referencing it to a human reference genome, the GRCH38, and then we looked at which genes are the most expressed. And the level of significance was 16, which means it had a log2 fold change of 4, and then we ranked them of how significant they were. And so in this battle, these genes are basically like the soldiers, and the most significant ones are the ones that have like the most stars, or like the super soldiers. And we wanted to see which ones are the super soldiers, and then see how many of them they are. So we wanted to visualize it and see compared to the total genes that are being expressed. And then we wanted to look at their functions, and if between Salmonella and Listeria, if they have certain functions that are the same. So this is our volcano plots, and volcano plots are basically giant scatter plots to look at drastic changes in the log2 fold changes. So you see this is this right here is the Listeria, and the green dots are the most significant ones, the super soldiers. And we found that 0.79% of all the total gene are the super soldiers, but Salmonella has 2.64%. That's so much more than this Listeria had. And then we also looked at if the Listeria and Salmonella had genes that were the same, and we found that 233, and these are the purple ones right there. Okay. And then we also looked at the data themselves. So we have Salmonella on the left and Listeria on the right, and we found we can see the top three with the highest log2 fold changes, which means they were most highly expressed. And Salmonella had the most expressed log fold change with 11.28, and you can see that they have the same number one gene, which is IL-12B. And then so this could be like the main gene that's like the most expressed. And we also looked at the molecular function using a gene ontology. And you can see like there's white boxes and like darker orange, and those are how important they are based off their function. Also like the cytokine signaling function. Cytokines are basically the messengers. So in a battle, there are people that have to talk to each other. So the genes, they talk to each other, and they use cytokines for that. And if you look at the red boxes, those are the most significant ones, and we see that the molecular function for both of these, it's a little small to read, but it's ion binding. So for Listeria and Salmonella, the general antibacterial response is ion binding, and for Salmonella there's also another red box, and it's for DNA binding. We're still looking if there's other diseases, if they also share that molecular function.